Here we'll discuss about the ionic composition of endolymph and perilymph. As we have discussed, endolymph is situated inside the scala media and perilymph is situated inside scala vestibuli and scala tympani. Check the composition of this perilymph, where the sodium concentration is more than potassium concentration, and the composition of ions is exactly like cerebrospinal fluid, which already we have discussed. Now coming to the composition of endolymph, which is present inside scala media. Here, because it is a closed chamber, the composition of the sodium and potassium is completely different from perilymph. Here we can see sodium is less compared to potassium. The potassium concentration is much high in case of endolymph. Now, from where these ions are coming? Because it is a closed chamber. These ions and the fluid are secreted by stria vascularis. This where the blood vessels are situated. This SV stands for stria vascularis. From these vessels, the ions of fluid is secreted and it is accumulated inside the scala media. Though the hair cells are bathed in endolymph, the basilar membrane is relatively permeable to perilymph of scala tympani, and consequently the tunnel of Corti and the bases of hair cell are bathed by perilymph. Points to be remembered: the inside scala media contained endolymph. The composition of the endolymph is different from the perilymph. The deep part of the hair cell is bathed by endolymph. The basal part of the hair cell is leaky to perilymph. So the same cell is coming in touch of both the fluid. Because of this and the difference in the composition of ions, creating a huge potential difference, which is responsible for the signal transduction. Now the endocochlear potential, what it is and how it is formed. The last slide we have discussed about the composition of endolymph and perilymph. There is a huge difference of potassium concentration. The scala media contains endolymph, where the potassium concentration is higher than the scala vestibuli and tympani, which contains perilymph. This scala media is electrically positive compared to the vestibuli and tympani because of the potassium concentration is too less in perilymph. Ultimately, this is creating an electrical gradient of about positive millivolt. That means the scala media is electrically positive compared to these two, scala vestibuli and tympani, because of the potassium concentration is too high compared to other. This electrical gradient of positivity, that is a positive 80 millivolt, is called endocochlear potential, and this endocochlear potential is responsible for the signal transduction and thus the audition. This electrical potential difference is responsible for the nerve stimulation at the synaptic level. The continuation of the last slide, how the endocochlear potential has been formed. Now, next, at the basolateral resting membrane potential of each auditory inner hair cell is minus 60 millivolt, and the endocochlear potential, as we know, it is minus 80 millivolt. And this potential gradient that induces the movement of the ions into the hair cell includes both resting membrane potential of the hair cell and the positive potential of the endolymph, that is the endocochlear potential. So together, this two potential becomes 140 millivolt. Thus, the total potential difference is 140 millivolt. Therefore, a change in the membrane conductance in the apical membranes of the hair cells result in a rapid current flow that produces the receptor potential in these cells. And this current flow can be recorded extracellularly as a cochlear microphonic potential or receptor potential. Lastly, we are going to discuss about the formation of receptor potential. In this two diagram, we are showing the hair cell, how it is getting depolarized and how it is getting hyperpolarized, in which situation. As we have discussed, then when the shorter hair cell moves towards the longer hair cells, that means the stereocilia, there is a stretch of tipplings, and this stretch of tipplings causes the opening of mechanosensitive cation channels. Now, the hair cell projects into the endolymph, that is the hairs or the stereocilia projects into the endolymph. At the base are bathed in perilymph. This arrangement is necessary for the production of the receptor potential, where already we know the potential difference is almost 140 millivolt across the cell membrane. That is, a gradient across the apical membrane of the hair cell is 140 millivolt. This electrical gradient difference between inside and outside causes huge entry of potassium from endolymph, that means from outside of the cell, through this mechanosensitive channel inside the cell. This positive influx causes depolarization of hair cell. Because of this depolarization, the voltage gated calcium channel which are present at the base of the hair cell, it gets open and causes huge influx of calcium inside the cell. These calcium are responsible for the release of neurotransmitter inside the synaptic cleft. Here in this case, it is an excitatory neurotransmitter, that is glutamate. After releasing glutamate into the synaptic cleft, it binds with the receptor and causes a receptor potential, which is excitatory end plate potential. So this the way the nerve signals are generated from the hair cell to the afferent nerve, and signals is summated and transmitted as action potential through the afferent nerve. Now opposite happen when the longer hair cells move towards the shorter hair cells. Because of the stretch or less now, the tipplings get relaxed or loose. So because of the stretch has been gone, now the mechanosensitive potassium channels are closed and there is no influx of potassium inside the cell, and cell becomes hyperpolarized. So the transmission of nerve signal stops. So ultimately we reached how the receptor potential is formed at the level of synapse.